Welcome to our seminar for this week. Due to popular demand from last week and trying to live up to the high standards that were set by Dr. Cal, who did the seminar series last time, we have orange juice this week. So for those of you who don't like the caffeine buzz, feel free to sugar buzz. Yeah, feel free to have it. Yeah, we still have the sugar. We still have the sweets here. Haven't gotten the mixed nuts out yet. But anyway, it's a pleasure to welcome our seminar speaker here this week. He hails from all the way across the river at Washington State University in Vancouver. Yeah, across the border. So it's Dr. Xu. He's from the Mechanical Engineering Department there at Washington State University. He has a background in thermal engineering from Tsinghua University in China, and then went to Columbia University for his master's and PhD work. He's been on the faculty at Washington State now for several years. I think this was finishing your second year, right?
Yeah.
And he's going to talk with us today about some of the research he's doing. He's a DARPA Young Faculty Award winner, and he's got some DARPA-funded research he'll tell you about. But if he gets too deep into the applications, he might have to lock the door and build some memories. But anyway, let's all welcome Dr. Xu. Thank you.
Thank you, Dave, for the introduction. Good morning, everyone. So before I get started, may I know how many of you are actually graduate students? Oh, nice. Good. So everybody can hear me. And may I know how many of you have actually taken any class in fluid mechanics? Ever? Great. So I hope my talk will be understandable to you guys. So this topic of my talk is about microfluidic manipulation of multiphase flow and a few potential applications that I'm currently working on. And this content is about a few projects that I've done in the past few years, and which was combining to this coherent theme for this talk. Specifically, I will talk about the generation, elimination, and also control of multiphase flow in microfluidics. I guess I can skip this slide. This is my background. And oh, by the way, this is the picture I took from the window of my office. So we just moved to a new engineering building, and I'm at the corner of the building, so I can see two directions. This is the mountain in St. Helens that I can see. And of course, when the weather is good, which is not the case for the most of the time. And this is the other side of my building. I can see part of the campus. And this building is actually the old engineering building. So our office moved to the new building, and my labs are still in this building. And the most part of the campus is actually this direction that you are looking at. And my lab currently hosts a postdoc, a few graduate students, a small army of undergraduate students, visiting student, high school student, etc. And the funding resource is mainly from DARPA and also some other small funds. So back to the research. You may wonder what is actually special in the microscale world, and what makes the multiphase flow an interesting topic of study in microscale world, right? And so first thing is scale is not nanoscale, so there's no quantum effects there, right? So the basic physics is actually the same as our macro scale. So the difference is that different physics play different roles at different scales. So let's take a look at this ratio. So if we take the ratio of surface forces to volume forces, as you can see, the surface force is proportional to surface area, and the volume force is proportional to the volume. And this makes the ratio inverse proportional to the dimension, right? So if still the scale goes smaller and smaller down to micro scale, and then this ratio becomes very big. That means surface force will dominate over volume force, right? That's what's special in microscale world, and that is why this little bug can work on this air-water interface, while this fat guy just sinks to the bottom of water, and while he does not even notice, oh, there's a water interface. Right? So multiphase flow in microfluidics is kind of a hot topic in recent years, I would say in recent 10 years. And various of applications have been explored already. For example, researchers have found that by squeezing different channel flows into another channel carried by oil, these little droplets, they can be used as chemical reactors. I would say these are the kind of world's smallest chemical, beakers. And the chemical reaction happens in small droplets. They have much faster rate, and they are highly sensitive, and also they require much less sample consumption because sometimes, the biological samples, they are very expensive, like proteins, etc. And also, they found that you can even, these little droplets can even carry solids. For example, this one is protein crystallization inside droplets. And another example shown here is that it has been found that we can use one droplet to encapsulate a biological cell. And the good thing about this is that if you lysis the cell, say using laser or electrical field, so the cell membrane will break, so all the content in the cell will be released, right? And the good thing is all the content will still be confined inside that droplet. Therefore, the biochemistry chemical is strong, so that is more detectable. And also, this droplet can be used to move this content for downstream further analysis. And these are micro droplets. Micro bubbles are also very useful. For example, in this case, it has been found that by squeezing a trend of gas bubbles through this channel, we can enhance heat and mass transfer. It is because there, there's a kind of strong vortices forming between gas bubbles. These vortices are very good at stirring fluids, so that heat and mass transfer can be enhanced. Another example shown here is by attaching a gas bubble to a rod, and then we can control the vibration of the bubble by using this piezo actuator attached to the bottom. So if the piezo is on, then this bubble is oscillating, and this oscillating bubble can attract things around it, so it can catch particles. So if we move this rod to another place and turn this off, then particles can be released. So it seems that there's various of applications have been explored for multiphase flow at microscale. So the first research question I asked was actually about the generation of droplets and bubbles in microscale. And before we take a look at actually how people generate drops and bubbles in microchannels, let's take a brief review of how people generate droplets in the atmosphere like 40, 50 years ago. This picture shows the old inkjet printing technology where a trend of droplets was formed, and then this droplet was charged and deflected so that unwanted droplets are sorted away. This technology works very well. It can be used to print stuff, right? However, the system is quite complicated, and especially this entire sorting system, right? So the upgrade of this technology is called inkjet printing technology. For example, in this case where we have a heater inside the ink, if we send a pulse of heat, a vapor bubble is going to push the droplet through the little hole to make one droplet. And this is called drop-on-demand generation. And this technology not only works well for printing, but also has brought up many fancy applications. For example, we can print optical fibers on demand, resistors on demand, adhesives on demand, and even aroma on demand. I actually like this one because this can be coupled to computer games. If you run into a rose garden, this machine can release, the smell so that you can feel that. So anyways, these are the technological paths of making droplets into the atmosphere. So now let's take a look at the state-of-art way to make droplets in the microfluidic chip. Basically, we're following the same technological trend. Today's microfluidics, they're very good at making a trend of droplets, either using this so-called T-junction, this is also another T-junction, or using this flow focusing device. That means we have water stream here, and this water stream is pushed by oil stream through this little hole and form droplets. These these technology works very well. As I already mentioned, there's all kinds of applications. However, if we want to, say, manipulate say if you want to take a look at this droplet, or if you want to move this to another place, or if you want to select droplets, then this technology is kind of annoying, right? Because you have a train of droplets. It's hard to do single manipulation. So my research question was, can we make single droplet on demand at a time? And this technology basically is enlightened from this the first two graphs, basically. And if we can make single droplet on demand, we can add flexibility and versatility to droplet microfluidics, right? So before that, I want to briefly show how we can actually make microfluidic chips in the lab. Basically, we have two ways. One is called soft lithography. This method was developed by Whiteside's group at Harvard in 1998. And this basically says if we can use the lithography technology, make microstructures on top of a wafer, and then we pour the so-called liquid PDMS material. And then when this material is cured, then we become solid. Then we peel it off. So we have microchannels on the soft rubber. This method is very good at making very small channels, for example, down to a couple of microns. And also the surface of the channel is very flat. And however, this method takes a graduate student like 2 weeks to make a chip, probably. And another way we explored is so-called micromilling. If you take a look at this movie, this end mill bit is about 100 micron size. It's about the size of your hair. And with this method, we can also make microchannels, microstructures. And the roughness is okay, resolution is acceptable. It cannot make super small channels, but it's good for about 50 micron to a few centimeters even. So these are the two ways we can make microchips in our lab. So let's come back to the drop on demand thing. So the chip I made to achieve the task of drop on demand is by making one horizontal channel filled with oil, and then by making this big blue chamber filled with water. And then I attach a piezoelectric actuator on top of this chamber. So the idea was if I send a pulse, electrical pulse, to this piezo, I hope I can push this water into this oil film so that I hope the breakup happens and the droplet can form. And let's look at this experiment. So it works. So if I send a pulse to the piezo, then at this nozzle, I can see a droplet forming. Right? And the characterization of this method shows that it's kind of amazing. By changing the nozzle size, shape, and also varying the pulse shape and the duration, we can have droplet produced one at a time over about 3 orders of magnitude, from a few picoliter to nanoliters, basically. And we can have kind of fine control of droplet size, basically. And then we went further to see how we can use this technology to do something. One thing we found is that if we put 2 nozzles facing each other, and then we make droplets at the same time, so 2 droplets will form in the center of the channel, and then they will mix and be transported, say, to a contraction where mixing is enhanced. This way we can do chemical reactions inside droplet. By using different size of pulse, we can make different size of droplet, so we can titrate the chemical reactions. And another thing we found that is if we keep making drops to the original drop, that is merged to previously made drop, we can control the droplet size in a digital way, right? Because we're adding one by one. This way may be coupled with the previously mentioned fine-tune of the droplet size to give a wider range of the droplet dispense. Another interesting movie we found is that in some cases, if one drop is made, and then there's a subsequent excursion of the meniscus, this at some situation can break up the drop so that we have two smaller droplets. And this never happened in the drop-on-demand technology in the atmosphere, because the droplet always just fly away, right? Because in the air. Here it's probably because it's filled with oil, right? So this droplet cannot go too far away, so that later can be broken up by the meniscus. And one of the frame here mimics one of the famous cartoon character, and there's actually anecdote. In the paper we submitted, we mentioned that we said this is a Mickey Mouse dispense, and the reviewer hate this. He said we should not say this kind of thing in the scientific paper. Then we say, okay, we change this sentence to one of the frame briefly mimic the famous cartoon character. So people know this is Mickey Mouse, basically. And we cite Disney publications. So, yeah. But we don't really know what's the application of this. It's kind of fun. Maybe in this way we can make even smaller drops, right? Okay. Then we take a look at deep into the application with relevance to biology, because as I mentioned, it's very useful to encapsulate biological cells in the drop. There are going to be many advantages for cell analysis. So we were wondering if our technology can be used to encapsulate cells. What we did is we put a bunch of cell-sized particles, say 10 or 20 micron, and then we shake the chamber gently. They move around. Once we see a droplet, a particle is near the nozzle, then we send a strong pulse to make a drop. And then we can make a drop that actually encapsulates the particle. So this demonstrates the ability of cell encapsulation. And by the way, I forgot to mention, all these movies are high-speed movies, so they actually slow down. And the rate is about kilohertz. That means you're looking at making 1,000 of these droplets in 1 second. So this is cell encapsulation. And we actually went one step further. We did some collaboration with a group who does radiologist study. What they did previously, before our collaboration, they put cells in a petri dish and then shoot the cells with, say, neutrons or electron beams to see how cells can be damaged, this kind of thing. And this way, there are many drawbacks. For example, they can only work with cells that can actually attach to the petri dish. And also the throughput is very low because you have to move the stage to the next cell and shoot the beam again, right? And also the chemical signals are kind of weak because whenever there's signal, it's diffused in the entire petri dish, right? By collaborating with us, we made a device that can actually encapsulate cells into droplets. And then we move the droplets in a stream of flow so that they calculate, when the drop going to arrive at this location, then they shoot the beam. And in this way, we brought many advantages to them. For example, now they can study cells that cannot adhere to the walls. And also the throughput is much higher because we send 1,000 droplets in 1 second. And also another added feature was they now can study the so-called bystander effect. That means they can put 2 cells in one droplet and shoot the beam to one cell and see how the other cell react, if he's happy or scared, this kind of thing. And this is kind of still ongoing project. And in the end, we were thinking we can make droplets. Can we make gas bubbles? We tried the same way. We tried to use a piezo here and push gas, but I found it did not actually work well. So we actually used the opposite way. So we put gas there on top of the channel, and the rest of the chamber filled with water. So instead of push, we use piezo to pour the liquid so that the gas can be poured into this nozzle, and then we have a breakup to form a gas bubble. So we can also use the similar technology to generate gas bubble on demand. We still don't know how to use this way, but it's kind of an interesting feature to have. So that's about the story of generation of multiphase flow in microfluidics. So the next part of my talk is about elimination of multiphase flow in microfluidics. You may wonder, since droplets and bubbles, they're so useful, right? They have so many functions, applications. Why would you want to remove them? There are a few reasons. For example, gas bubbles or drops, they can clog the channels. Especially you have a corner of channels, they might get stuck at the corners. And it also seems that if you have water stream contain gas bubbles, then this dynamic performance can be affected. Also, in some cases, if you have a fuel cell, then some of the product are in gas form, and these gas bubbles need to be removed as quickly as possible so that the reaction can keep going. Right? And then this slide basically says that what are the current ways that we use to remove gas bubbles from a microfluidic system. One way they found is that if you make this spiral-shaped structure in this channel, so that when the flow containing bubble flow over this, then the bubble going to be concentrated in the center line, and then they put a tube here so that the gas bubbles can be collected and removed away. This technology worked, but it's kind of super complicated, and also some water can also be leaked through this tube. Right? And another way has been developed is by putting a PDMS membrane on top of this chamber. Basically, PDMS is a soft rubber-like thing, and also it's gas permeable. So that means gas can actually diffuse through this soft rubber membrane very slowly. So if we have a gas bubble in a chamber, after, I don't know, an hour, then this bubble is gone. This method works, right? But you can see it just super slow. It takes an hour to remove this small gas bubble. And another way to do gas-liquid separation is by using this kind of porous structure, by using these little channels and make them hydrophilic. That means water likes the surface. So if you have a stream of water gas flow comes in, the liquid likes the surface here, so the liquid gets sucked into this surface. However, gas cannot move into this because of the capillary forces. So you have liquid here and gas here. This method actually works very well. It just it's kind of complicated in terms of the microfabrication process. What we did is actually by using a porous membrane to actually achieve the similar effect that is shown here. So the idea is that if we can put a porous membrane somehow to the one side of one wall of the channel, so that one phase can go through this membrane, and the other phase can be prevented to go through this membrane by capillary forces. And if this membrane, porous membrane, is happen to be hydrophobic, then the phase that can actually go through will be gas, and the phase will be kept in the channel will be liquid. This method is actually not new. What our contribution is, we studied the physics and also came up with the design criteria for it. This is what we did. We used the micro milling method. We made a channel, and then we push gas here, so we have a stream of gas bubbles. And then we put a porous membrane on top of this chamber, just tape it, kind of simple. And we have all kinds of porous sizes tested. And we have a movie, actually, showing that. So in this case, in this movie, the bottom wall of this chamber is porous membrane. As you can see, the gas bubbles comes in, they get smaller and smaller, and eventually they get sucked away completely. Right? One interesting we found is that at almost the end stage of the bubble, we had all kinds of shapes of the bubbles, even like this one. And interestingly, this transition, from this almost spherical shape to this sharp, triangular shape, is happened around Weber number equal 1. That means it kind of makes sense, right? If you understand what Weber number is, this basically compares the inertial force to surface tension force. So if this inertial force is too strong, then surface tension is not strong enough to hold the shape. So at higher Weber number, we have this sharp shape. Okay. This is just some interesting to explore. So we come back to the physics of this gas extraction using porous membrane. So we wanted to define a successful gas-liquid separation. What we want is the gas bubble to be completely extracted, right? And we also do not want any liquid to leak through the membrane. And we were thinking, what are the conditions are needed to ensure this successful gas-liquid separation? So the first criteria we found is that the size of the bubble need to be bigger than the channel size, because you need the bubble to be in contact to the porous membrane so that it can be extracted. This is just a criteria. We can actually easily get around it. In a practical case, we make a contraction or some obstacle or something, right? And the second criteria, during the development, we think that we need to look at the gas transport of the porous membrane, because it takes some time for the gas to completely transport over the porous membrane, right? This will become our second criteria. It seems that by using this Darcy's law, basically this law says that the liquid flow, the gas flow over the porous membrane is proportional to the transmembrane pressure. That means the pressure difference across the membrane. It's kind of simple, right? And this gives us a clue that you need sufficient time for the gas to be extracted, right? So this becomes our second criteria. That means the membrane needs to be long enough so that the bubble has enough time to go through the membrane. And this model actually works very well for the smaller pores. For the larger pore size, it seems that it doesn't really work well. So we looked into that. What we found is that sometimes if the gas bubble is too fast, there's going to be a liquid film in between the bubble and the porous membrane. So if you have liquid film, then the gas, there's no way to be transported through the porous membrane, right? Because the liquid film impedes mass transfer. And what we found that if you look at this little drawing, if we have gas bubble moving in this direction, this is water, and this angle is called receding contact angle, right? When you measure contact angle, you measure in water, right? And this receding contact angle actually is a function of velocity. That means if you have faster and faster moving interface, you're going to have a smaller and a smaller receding contact angle. And once this receding contact angle reaches 0, what happens is there's going to be a liquid film deposited on top of the membrane. In that case, you will have no gas transport at all. And by using these criteria, using this physics, we found that if you want to have successful gas extraction, the velocity of the bubble needs to be smaller than a critical velocity to prevent the formation of the film. Okay, so with the previous three criteria, we can make sure all the gas bubbles can be extracted. So the last criteria is quite simple. We do not want water to leak through the membrane. And this leakage of water through the porous membrane is because of the pressure is too high. If the pressure is too high, then the surface tension cannot hold the pressure anymore. So the last criteria is quite simple. So the pressure needs to be smaller than the critical pressure that the pore can hold, actually. So to summarize this part, if we want to have a successful gas extraction, the bubble needs to be bigger than the channel size, and the porous membrane needs to be long enough so that bubble have enough time to get extracted. And the third is the bubble needs to be slow enough, not go over a critical speed so that there's no water film forming. The last is the pressure cannot be too high so that water, cannot leak through the membrane. That's what I've done for the elimination of multiphase flow. Everything's okay so far? So the last part of my talk is about control of multiphase flow. And in terms of control, there are many ways to control: electrical, geometrical, magnetical method. What we specifically interested in is we are interested in the interaction of the multiphase interface and ultrasound. Because if you put an ultrasound in the flow field, then there's lots of interesting phenomena around the interfaces. For example, I'm going to talk about these three things. In an ultrasound field, around the gas-liquid surface, we can talk about the interfacial waves, and also there's so-called Bikini Sauce. I think that's the pronunciation. I'm not sure. Bikini Sauce. And also there's a microstreaming phenomena. And I'm going to talk about some phenomena, some science, hopefully, and also potential applications. So the first thing is the interfacial waves. What we did was actually quite simple. This discovery is kind of serendipitous. So what we did is we had a microchannel. We put gas here. We put liquid here. So we have liquid-gas interface, right? And then we put a piezoelectrical actuator somewhere in the chip. It's actually buried in the chip somewhere. Then we turn on the piezo and adjust the frequency. It's kind of obvious the interface is going to be shake. It's oscillating, right? It has found that if the intensity of the piezo is low, meaning the voltage applied to the piezo is low, then there's going to be these traveling waves. You can see, right? These are traveling waves because they move from the two sides to the center. And this happens, when the intensity is low, and the frequency of these waves are the same as the excitation frequency. There's not much, exciting things there. However, what's exciting is if we turn up if we increase the intensity of the piezo by increasing the voltage, at some point we found that standing wave is going to happen there. As you can see, this finger-like wave. If you just focus on one of the wave, it's going up and down. So it's standing wave. It's not moving around. Right? And this happens. So in most cases, this wave, the frequency of this wave is half of the excitation frequency. And we look at the literature and see, oh, this is reported, and it's actually named after Faraday. So this is a typical Faraday wave. The Faraday wave happens when you have a, say, tank of water, and you just shake it, At frequency F, they're going to be half of the frequency wave happens at the water-air interface. This is called Faraday wave. And we also found that at some point, the wave frequency is the same as the excitation frequency. It's also not very exciting. However, what we found is that at some other cases, the frequency of the wave can be even higher than the excitation frequency. And actually, to be exact, it's 1.5 times. And this is kind of weird. So we went through the literature and found some previous, report of this kind of evidence, especially when people do the ultrasonic imaging. They report that they found that the microbubbles sometimes oscillate at an even higher frequency than the incoming beam. But nobody has actually ever observed this wave, directly. And this is so-called superharmonic wave. So we look a little bit deeper into this phenomena, and we took a look at the wavelength and the frequency relationship. It matches the typical dispersion equation. This basically can be found in any classic fluid mechanics books. And for the standing wave, because it has half frequency and also 1.5 of the frequency, there must be some nonlinear effects there, right? What we thought it's the source of the nonlinear effects. What we thought it might be coming from the changing gap, because we have a piezo, we have a chamber, right? The gap of piezo, the gap of the chamber might change, and then this meniscus gonna be deformed in a different curvature. This may be cause the change in the surface tension force. So we actually consider that this effect, the surface tension change, and we put in this effect into the dispersion equation, and then we come up with this equation. I'm not going to talk about too much detail about this equation. The idea is that this is a linear oscillation equation with oscillating coefficient, right? And this equation is called Matthew equation. And this Matthew equation, the stability of this Matthew equation tells us that the wave can actually oscillate at half of the frequency, the same frequency, and even 1.5 frequency of the excitation frequency. So everything can be explained using this equation, and this is actually called parametric study. One interesting thing is that the mathematics actually tells us that there are even higher, bands, higher nonlinear tones. However, in the experiment, we can only observe up to this one, 1.5. Maybe because when you go higher and higher, maybe the viscous force will damp the wave or something. So that's the kind of the story of the waves, and we don't really know what's the application of these waves. It's just kind of a cool thing, and it's kind of the side project that we found, and about this superharmonic wave. Let me see the time. So the second effect I mentioned about oscillating bubbles is called this Bikini Sauce, this one. Right? This force is because of the scattering of the gas bubbles in the ultrasound field. And this force is a function of all these parameters. Basically, it depends on the density of the particle, of the surrounding fluid, size of the particle, and oscillation of the bubble. What this equation basically says that if you have a gas bubble oscillating, and the things, in the surrounding fluid will feel a force coming from the bubble. It's either attractive or repulsive, depending on the density, right? And this force actually has been reported, and they've been using this force to, track particles. If you remember, one of the few first slides I showed people using oscillating bubbles to trap some particles and release it at the other place, right? What we did is that we put some worms. These worms are called C. elegans, because the worms are about 500 micron up to 1 millimeter, and these bubbles can be made easily with the size about 500 micron. We just want to see how a worm will react, kind of exploratory. And the reason we are interested in worms is actually because these C. elegans kind of widely used in the biological studies. They are used as model animals for many, many things. And one of the reasons is that this is the simplest animal that has the neuron system. It only has 956 cells, something like that, if I remember correctly. So we know the details of each single cell in this animal, and so that's why this worm is widely used for many, many studies, especially for neuron part of study. And in these studies, scientists, they always want to, select a worm, transport worm, immobilize worm, right? So the idea was by using oscillating bubble, maybe we can achieve, worm selection, transport, immobilization. So that's the idea. So let's look at this movie. So these are gas bubbles, 2 gas bubbles. This is a worm. So as you can see, the worm was attracted and pulled directly to this oscillating bubble, right? So what we have demonstrated that these oscillating bubbles can trap worms. And using this feature, what we thought was maybe we can do elegans, these worms, enrichment. We made a slightly different geometry. In this case, we cut a channel, and we put an array of holes. By filling water on top of these holes, the gas bubble is going to be trapped inside, right? And then we flow the stream of water that has worms inside, and we oscillate these bubbles so that these bubbles can trap the worms, and liquid can flow through. By using this method, we can actually enrich the concentration of the worms in the liquid. What we found is that depending on the flow rate, we have a certain efficiency of trapping, but it's all about 0.789. That means we can trap almost 80% of the worms in the stream. And also we found that by running the stream for 30 seconds, we're going to have about 5-6 times more worms in the same volume. So we did elegans enrichment using oscillating bubbles, basically. Another interesting thing we found is still preliminary results. This movie just obtained a few days ago by my student. What we found is these are two oscillating bubbles, right? And we have big worms, small worms trapped to the same bubble. And then this big worm can be released because he might be the strongest one there. So this tells us that the stronger worm may have the ability to escape from the force. So my student is still doing the test, try to use this to achieve worm selection. What our plan was by attracting all sides of worms around the bubble. If we lower the voltage gradually, we think the strongest worm gonna escape first, and then the weaker form, worm. That means the smaller worms. In this way, we wanted to achieve worm separation. We also found that by using oscillating bubble array, we can manipulate single worms. For example, in this case, we have one worm moving from this bubble to this bubble. While the worm is trapped, he actually moves around the bubble, rotates the bubble. What we found is that if the worm is pointing to one direction that we want the worm to go, we just release it, just turn off the piezo, so that it can swim to that direction. And then we trap it again. And then we release it again until it goes to the right direction, and then we release it. So you can see we modify the pathway of the worm. This is just a square. Maybe we can use we can do more fancy thing. Maybe we can do triangle PSU. Yes?
Is this switching done by human observation controlling it, or is it something that you've automated?
It's not automated, just by observing. And my student, look at this on the computer screen, and then turn it on and off. So worms are directed. Yes?
The scary idea is, how do we know or not that the government is only doing that to us?
Huh?
It's the tax
turning on a stimulus. You're going the wrong direction. You need to be going to work today, right?
Yeah, that's the idea. Yeah, because these. So. Yeah. The worms actually, when they swim a little bit longer, they actually can turn or something. That's why we cannot. That's why we need to have an array to keep adjusting the worm. So if you want the worm to go exactly from here to here, so you might want to whenever it's kind of deviate a little bit, you pull it back. Deviate a little bit, you pull it back. So. Yeah, this kind of just a fun thing for my student to do, and we. Yeah.
What kind of signal are you using? The piezo that's activating that? Is it a repetition? A pulse?
Yeah, sine wave actually. Yeah.
What's our frequency range?
This range is in the ultrasound. We use about 100 kilohertz also, and we think it's because of the natural frequency of the bubble. Yeah, there's complicated equations for even a spherical bubble, you know, but it's about in the same range. Say this is about 300 micron, and the natural frequency is about, you know, 2 or 3 kilohertz. Yeah.
So if you raise the voltage, does it raise the attractive force?
Yeah. If yeah.
Just like not identically, but
Stronger. Yeah.
okay.
Yeah. Yes.
Are all the holes the same size? I was wondering if you had different sizes, that would imply like they have different frequencies, so you'd be able to.
That's a very good.
manipulate.
Yeah. That's a very good point. That's a very good question. We tried. It did not work.
Oh, it didn't work.
Yeah, it did not. We don't know why. It's weird. Yeah, we had decided. Actually, as you can see, these bubbles are not the same size. They have, you know, they have smaller and smaller size, but we found that they actually oscillate at the same frequency. I just have no idea. Maybe this is forced vibration. Maybe to have the, you know, to have frequency selectivity. Maybe we have to use the, you know, the actual natural frequency, so that other bubbles, they do not vibrate at all, right? That's what I thought. We actually have no clue why.
Could there be a material consideration?
Could be.
Using different materials or different fluids?
Could be. Now we were using aluminum because it's kind of, you know, solid and the sound wave transmits well. And then we use the ultrasonic gel to glue the piezo to the aluminum. It's good at transmitting sound. Yeah. We could try, say, silicon, plastic, all kinds of things. Yeah. It's still ongoing. No more questions so far? Okay. So these about worms, actually. And that's the last slide about this Bikini force, attractive force of the particles around the bubble. So the last phenomena I wanted to mention is so-called microstreaming. So this is kind of the same thing as before. We have gas here, liquid here. By oscillate the entire thing, we can obtain the fast, strong vortex thing in the liquid. And what we observe is that this flow speed is kind of second order to the voltage or the intensity, and this streaming kind of have some kind of resonancy frequencies. And this frequency, this phenomena have actually been used for several types of applications. For example, it has been found that by, you know, trapping this gas bubble in this side channel, the streaming can actually push liquid forward so that they can actually make a pump. And then in this case, by, you know, using these sideward trapped bubbles, when this is off, then turn this on, and mixing can occur. Right? You might know, you know, mixing is one of the biggest problems in microfluidics because of the small scale, and it's extremely hard to generate turbulent flow. Right? The Reynolds number is very low. Right? So this streaming serves a good way to generate, you know, crazy flow at micro scale, basically. And also, we previously found that if we attach something, rotor on top of the bubble, this can actually rotate like a micro rotor, like helicopter-like thing. The turning speed can reach as high as like 600 RPM, and we also estimate the power. It's about femtowatt range, but we did not do any further study. What we thought, maybe we can arrange this, you know, in some way so that we can extract power from this, maybe. But I'm not going to talk into detail about this thing. What I plan to talk about this microstreaming today is actually we found that we can use this microstreaming to do some study for hearing loss. So just a few slides about the hearing loss. Hearing loss is a very serious social problem. It's actually the number 3 public health issue. And this hearing loss is even worse in the armies. This is the number 1 disability of the soldiers from the wars. So the Army, especially DARPA, they're really interested in technologies to help protect hearing and this kind of thing. The reason might be the soldier, they hear the gunshots, explosives all the time, so that may be the reason, right? So a little bit deeper into hearing. Hearing that happens in the auditory system is because if we have sound wave comes in and this vibrates this membrane, and on this membrane we have this special cell which called hair cell. That means hair cells with hairs, right? And these hairs, they can bend, because we have vibration. If they bend and they have longer hair and shorter hair, they are kind of connected in this way. If they bend, then the ion channel going to open so that ions can go in and out, so that this electrical signal can be picked up by the brain. So in some way, this is our mechano-electrical transducer. It transduces mechanical vibration into electrical signal, basically. And this basically explains how we do the frequency selective excitation in our ear. We have basically a membrane, have different natural frequencies at different locations. So hair cells are on top of this membrane. If a certain frequency of wave comes in, then this part of hair cell is going to be excited, right? So the problem is that the functioning hair cells are not easily accessible because they're deep in the ear, right? If you take it out, it will not function. And if you want to develop drugs, you want to see how in the end, you want to see if the function can be restored, if the function can be damaged, right? So what I propose to DARPA is I propose to put hair cells in the microchannel, keep them happy alive with nutrition control, and use electrodes to detect the electrical signal. And one of the important part I proposed was the one you mentioned, actually. By using different natural frequency of bubbles, we can actually selectively excite microstreaming. And this microstreaming can be used to excite the hair cells. And then we pick up the signal, so we know, oh, this is what frequency, right? We can use this maybe to hear music or something. And that's the idea, basically. It's not working well yet, still under development. So the short-term goal is by developing this so-called ear on a chip. It's essentially an ear, right? It's hearing different frequencies of sound using hair cells. So the short-term goal is we can do drug screening, because there are all kinds of drugs. We want to know if they are harmful, or they are harmful to hair cells, or they are protective to hair cells. This needs a lot of screening capabilities. In microfluidics, it's very easy to control concentration of drugs, so that we can see if the function of hair cells can be affected. Long-term goals of this ear chip, I have a few in my mind. The most important thing, of course, we want to understand hearing, because the hearing, actually, the molecular mechanism of hearing is still not yet known fully. I think this is the last sense of a human, has not been understood clearly yet. And since I'm an engineer, besides this biological, medical stuff, I'm also interested in crazy things, right? For example, maybe we can make prosthetic ear, maybe not only use this thing to fix our hearing loss, maybe we can enhance our hearing. For example, maybe we can hear ultrasound, right? And we never feel about this thing, right? Ultrasound. Also, maybe can we use this thing to make maybe very sensitive biohydrophone, maybe? Also, in future, can we harness energy from sound? Because these hair cells, they turn mechanical vibration into electrical energy, right? All these crazy things maybe can be done in future. And let me see, I still have maybe 5 minutes. So the place we harness, we obtain our hair cells is actually from fish. Fish are a very interesting animal because they use hair cells along this lateral line and also the other side. They sense water flow around it. It's the same mechanism we use to hear sound. It's all because of flow, vibration, the mechanical signal to bend the hair. so that it can sense. So we just use hair cells from a fish lateral line, and then put in the channel and see if we can pick up some electrical signal. And this is really preliminary. This is a zebra fish that my student got, and we ping this fish and use some water flow to stimulate the hair cells, and we got some signal, but nothing can be understood at this stage. I think this is just purely noise. My student got this like several weeks ago. We have no idea what this thing is. Yeah, that's about the hearing study using multiphase flow, basically. So this is acknowledgement, and thanks for your attention. I welcome any questions.
So in one of your slides, you were showing some video of droplets that you were making.
Yeah.
The controlled droplet or droplet on demand.
Yeah.
In the video, there were some really small droplets that came just about every single time. Is that pretty consistent and uniform? Is that something you might be able to comment on?
Small droplet, right? Satellite droplet. Yeah, this is actually one of the biggest problems in the inkjet printing community. They want to really remove the satellite droplets. I'm sure I also had some of this problem. Maybe this one, I guess. There's little droplets, right?
It was a video where the
That's
droplet
right.
was moving in a vertical direction.
This one? Let me see.
There.
Oh, this one.
Even that one, or similar to that.
Yeah, that's right. So this satellite droplets happens all the time, kind of repeatably.
Yeah.
So yeah, this can be removed by fine-tune the pulse, basically. And this is one of the common problems. There is a problem in the inkjet printing community because these small satellite droplets, they affect the printing quality. But here we did not take deep look into that. Yeah, but that's a good observation. Yeah. Yes?
Which of the mixers holds the most promise?
Mixer, right?
Yeah.
In microchannel. There are a few things, people want to have fast mixing. They want the mixing to be super fast, of course. In that case, I know some researchers, they push the stream of liquid they want to mix to a very thin, to a very thin sheet of stream by pushing, from two sides. So that the scale are really small, even down to a nanometer. So diffusion happens under 1 millisecond. So it's in the microsecond range. But that case, it's super fast, but it's hard to detect the product.
I'm interested in
Oh,
the slide where you have
yeah?
the
Mixing.
bubble and the helicopter, and then you
Yeah.
have two other micro mixers.
This mixer produced from Microstreaming, they can do
So which of those is the better mixer?
this.
There's just one mixer then.
One mixer, yeah.
Just one mixer.
Yeah. This one can reach about 5-7 millisecond mixing time. It's not super fast. But the good thing is you can actually have large area for you to record image, fluorescent intensity.
And do you guys use this micro pump from Lee, or do you have a different mechanism, a different?
We did not use this as pump actually. In our study, whenever we want to pump something, we just use syringe pump. You can program the pump.
Okay.
And this on-chip pump still under development, I would say, because it's not working repeatably, and it also depends on the specific frequency. It seems to be, yeah.
On the previous slide, or just one before, where you had the ultrasonic
Yeah.
effect,
Yeah.
where you had
sound.
Oh, you had a little ultrasonic detector on one end that you were driving, and it had the 3x6 array.
Oh, that's right. That's the device we just made for the warmers.
Yeah.
Yeah.
Have you looked at putting like pairs of the piezo transducers
Oh,
and then
two?
driving them differentially, and then another pair opposite, and then varying the phase between them, because that will generate standing waves that you might be able to generate some little channels to move the guys along or something?
That's right. That makes sense. Yeah.
Are you familiar with DDS at all?
DDS?
It's a direct digital synthesis. It's a type of way of generating frequencies and phases. There are all sorts of chips available doing it. It's really
Not really.
so.
That'd be great. Yeah. That idea sounds interesting. And by varying the phase, we can actually control the
you'll get standing
location.
waves,
Yeah.
and there'll be troughs and peaks, and you can just move the little fellows around with it.
That's right. That's a very interesting idea. Yeah. Yes?
So on your hearing, I understand how you're stimulating the cells, but what exactly are you measuring, or what do you intend to measure on them?
The iron concentration change, but it's also proven to be extremely difficult. Initially, we thought maybe just stick an electrode there, it should get some signal, but not really. The signal is too weak. Now we have to look into the amplifiers and also some way to, I'm not sure if you are familiar with the patch clamp technology. So we really want to bring the electrodes in the capillary very close to the hairs, and we're still trying to do that. It's not easy.
Usually the patch clamp system I've seen are surrounded in like mesh, like a copper mesh, and very sensitive to all the outside.
Yeah. There's all kinds of things, and also the amplifier, filter, and the software. And we have to do very good, careful selection of these things. Yeah.
A dumb question: can the hair cells in that apparatus be considered to be still alive? Are they being supported by a biological process, or are they extracted and turned into a mechanical device?
We want to. They actually has to be alive, but we are still looking at ways to keep them alive,
Right.
and it's also not easy. What we have for now is we can use some drugs for the fish. They are lava fish. They are about like 2 millimeter size. We make them, immobilize, basically sleep, and then the hair cell is still alive. So we kind of have to use live fish at this point. And the problem is in this community, hearing community, there's still no good hair cell line. That means you can just buy. Still no such thing yet. There are some, but they're not functioning well. So, yeah. There may be some way to do in future. I've seen some paper. They already use stem cell. That's kind of out of my arena. They use stem cell. They program stem cells so that they become hair cells. I don't know how they did that, but maybe in future we can use this way. Yeah. The idea is try to integrate them in the mechanical system. Yeah.
Any other questions? All right, well, let's thank our speaker again.
Thank you.